me show you how to be a good Bayesian. Change your predictions.
In 2000, you and your father, so Donald Berry, you founded Berry Consultants. So what was the founding vision, and how has it evolved over time? Because we're 25 years later now.
Yeah, so in 2000, I'd worked on several design projects, and I very much was trained as a Bayesian, went to Carnegie Mellon. My advisor, Jay Cadane, is very much a Bayesian. So and this was right at the advent of Markov chain Monte Carlo, and being able to actually do computation in this, and very much enjoyed the modeling part of it, but I think as a Bayesian. And the interesting part of the world of biostatistics, the world of clinical trials, is at that time, and even now, it's a very frequentist dominated world. And you could almost say it's the last bastion of frequentist. It's kind of if frequentist is going to be successful, that's where it's going to be successful. It's set up for it and all of that. I think that's coming down, and we're finding that they're becoming much more complicated, much more interesting. A clinical trial is not just the data in the clinical trial, which frequentist needs that for its inference, and it needs this sort of closed space, and that whole world is changing. But in 2000, incredibly dominated by the frequentist world, and I think it's limiting in the trial designs, actually. It is strangely taking this sort of approach, limits the adaptations, limits the design. And you can imagine when I tell people what I do that aren't statisticians or aren't in this world, is we take what the old model was of a trial where you say, go enroll 200 patients, and then in 3 years you look at the data, and invariably you say, oh shoot, I wish we'd have done a different trial. I wish we'd had a bigger sample size, a smaller sample size. We'd have changed the randomization. We'd have changed the patients. There's a whole aspect to this, that the idea that you run this trial for 2 or 3 years, and only then do you get insights into the data, they look at you like, that really is what's happening? So we thought the idea that you do multiple interims during the trial, you make sure it's progressing the right way, you allow it to change to answer the relevant questions, are better trials, better use of patients, better answers. And it was really jumping in 2000, having no idea whether this would stick. And, all of a sudden this is my way of income, and have three small children, but was willing to take the risks and go into this. And, hence you talked about 30 people, and now, there's a great deal of innovation in this space, but it was initially a lot of medical device trials, which has a certain synergy to Bayesian, to then, drugs, within that situation, jumping on and really doing that. So it is something that hit a sweet spot, and there was a great deal of interest, and still is today, in alternative, called novel approaches to clinical trials.
Yeah. That makes sense. And so actually, before diving a bit more into that, can you give listeners an idea of what that means concretely? Like, what does your job look like? We're on a Tuesday in the morning, so right now you're recording, but usually what do you do Tuesdays at 10 a.m.? What does the work you're doing look like concretely?
Yeah. So it's typically that we are working with an organization that's interested in designing a trial. And that organization can be a pharmaceutical company, could be a medical device company, it could be the NIH, it could be a federal government, it could be a patient organization, where they're interested in a trial design. They're interested in a question, or usually multiple questions, and they want to design an experiment to answer those questions. So we will work with them on these designs. And so, for example, there may be a particular drug, and they want to get it approval by the FDA, and they want to run a trial to do this. but they have questions. Maybe there's 3 potential doses and they don't know the right dose. Can we run a trial where we explore the 3 doses, answer the question, figure out the right dose, have it advance into a confirmatory stage, enrolling against a placebo, and that demonstrates safety and efficacy in the same trial? Maybe even a question about, do we have the right patients? So the alternative would be you run a trial to investigate dose, you wait a year, you analyze the data, you run another trial to then compare it. Can we do this in one? Can we do this more efficiently? That might be a standard, and we work with them on the trial design, what that looks like. We're simulating different trial designs, comparing them, building a better design, and not infrequently within those designs, the thing that's driving those, the dose decisions, the sample size decisions, our Bayesian models are driving those decisions. And so we are building trial designs for a wide range of groups interested in building really a smarter, more efficient design than a simple fixed, let's look at the data in 2 to 5 years.
Yes, and actually, so I think it's a good segue to explain what the main issues are with the classic way of designing trials. You've mentioned already a couple times that there is, well, the problem of peaking, the problem of how to determine the sample size you need to get to the power that you want, things like that. So can you give listeners a rundown of how these things are usually done? And I'm guessing a lot of listeners will also be familiar with these terms, not only if they work in biostats, but also if they work like in any field where you do a flavor of A/B testing, where you have a placebo and you are testing if a treatment is more efficient than the placebo. Well, you're doing basically what Scott is gonna talk about right now. So maybe give us the rundown of what the current way, the classical way of doing things is, and where the main pitfalls and problems are, and then we'll enter into how you guys solve that and how Bayes is actually helping a lot in it.
Mm-hmm. So now trials there's a huge range of them, and they're typically grouped in phases. Where phase 1, you're really worried about safety. You're giving a new novel drug and it might be first in human kind of stuff. Through phase 2, where you're really trying to use multiple doses, frequencies, patients. It's more learning about it. To phase 3, which is confirming safety and efficacy. So all of this can be different, but the old model, and it sort of ties to a frequentist approach, is you enroll some number of patients on a control. It might be a placebo or on a control, and then some number of patients on a treatment. And you're addressing the question of, are patients doing better on treatment than they are on control? And yes, many experiments are set up in the same particular way. The frequentist approach to this is asking, typically, at using a p-value and doing a hypothesis test of, testing, are these treatments the same, or are we seeing statistically significant improvements in the treatment arm? So if you think about that, the p-value saying, assuming that there's no difference, what's the probability we see as big an advantage in the data as we see? And this could be time of survival in an oncology trial. It could be weight loss. What's the amount of weight loss in a weight loss treatment? It could be the rate of decline in Alzheimer's. It could be the neurological status in stroke. So it's creating these measures we're looking at, and are patients better off within that? Now, if you think about the frequentist approach and the p-value, it's okay, assuming some hypothesis, what's the probability of data in this experiment as extreme as it is? Now, this is actually an ideal case for frequentist. I think like Bayesian and all, because we write down the design. and we prospectively do everything. And so when we say, what's the probability this experiment would create data like that, we can actually calculate that reasonably well because it's all prospective. If you're out there doing experimentation out in many circumstances, you don't have a design. P-values are really hard in this thing. So this is a case where it actually applies really well. Now, there's two things that this starts to break down. One is we start to make the experiment more complicated, where there's multiple tentacles to this. It could go down multiple routes. We could increase the dose in the middle of the trial. We could do response adaptive randomization. There's the trial itself is becoming more complicated. So if you say in the frequentist, what's the probability we see data as extreme or more extreme? That's a sample space thing. And that's really hard if there's lots of moving parts to the design. So it becomes really hard to be a frequentist when the design gets complicated. So if you have to be a frequentist, it's hard to make a complicated design. You're sort of stuck with simple analytical things, which is really limiting. The other thing that's changing in the world of experimentation, clinical trials, is we don't just analyze the data in a trial in many circumstances. We're getting incredible amounts of data external to the trial. So if you start to say, what's the probability of getting data in my experiment as extreme as it is, it's sort of not defined very well anymore because we're using multiple sources of data and all of that. It's much easier to be a Bayesian in these circumstances because you only condition on the data you've seen and you don't have to worry about all the things that didn't happen in the world. So it's something that with more complicated design or data external to the trial, it's really hard actually to be a frequentist, where 25 years ago with simple designs, it was quite easy.
Okay, yeah, that's super interesting because I was aware of, well, these classical issues that you have in the frequentist framework where basically one of the main issues is peaking. If you look at your test results before you reach the sample size that you wanted to reach the statistical significance, then you increase the probability of false positive. Also, the issue of multiple comparisons. This is very hard to do in the frequentist framework. All these problems don't really appear in the Bayesian world because, well, you're not estimating the probability of the data knowing H0, you're estimating the probability of H0 knowing the data. And so looking like peaking is not really a problem, you're just updating your posterior.
Right. Now, by the way, this is the part you brought up, Frank Harrell. So if you go to In the Interim, I do a podcast with Frank Harrell, and you're welcome to. So we're both very strongly Bayesian, but this is the part where as two Bayesians we have sort of the conflict is, yes, what's beautiful in an adaptive design is if I do an interim analysis and then I get to a final analysis, that interim analysis doesn't affect the final. Looking at the data doesn't affect that analysis, and there's a beauty to that in the Bayesian approach where it does in the frequentist approach. In that, now we live in a world where people are interested in the experiment you're running. What's the chance that experiment creates a false positive? Now that's a place where Frank thinks we shouldn't even calculate that. But the world still is interested in that. In evaluating a design, you're interested in, does it get the right answer? Frequentist characterization of if you ran this design multiple times is not an irrelevant thing to evaluate a design. Now, it is when you're analyzing the data, and we agree on that, but there's still sometimes you still have to characterize the look. So there's no free lunch to looking all the time and finally saying, yes, we're successful in this world still.
Yeah. Thanks. This is a very important nuance. And so yeah, what I was saying also, my main thought was, okay, so I'm aware of these issues, these differences where, and I appreciate what you're saying, which is, well, this is interesting because in theory, that would be paradise for frequentist methods. But actually, de facto, it's not because of all these theoretical issues, picking multiple comparisons, things like that we just talked about. But also, what I wasn't aware of is what you were saying, which is, well, we actually have other data that's coming from outside our experiment. And so now the idea of if we repeated this experiment multiple times becomes very blurry because, well, are you able to repeat actually the same experiment? This is more and more doubtful. And so, yeah, that is actually a very important point I wasn't aware of.
Yeah.
And
Yeah.
so here I think it's interesting to actually try and get an example for listeners. So, because you've been involved in designing a lot of Bayesian adaptive clinical trials, and you also wrote platform trials. So, could you first define for people what these are? What's an adaptive clinical trial? What is a platform trial? And then walk us through a recent example that you found, especially in illuminating and what made it interesting. You see, like, was it its complexity, regulatory challenges, technological innovation, things like that?
Yeah. So an adaptive trial is one where during the course of a trial, an analysis of the data alters important parts of the trial. And I don't want to say it changes the trial because from the beginning you design it that way. You design it that at 50 patients, we're going to do an analysis of the data, and certain characteristics in that data will change the trial. We stop enrolling the low dose if it appears to be ineffective. We drop a patient population if it appears the drug's not benefiting those patient populations. And models will drive those decisions. So it's a trial that during the course of the trial, analyses of the data in the trial can change important characteristics. The patients enrolled, the arms given, the ratios of them, whether you keep enrolling, whether you stop enrolling, shifting to a new trial. There's lots of things to that. In some ways, a way to explain an adaptive trial is it's not one that you set it up, you run, and you only analyze the data at the end within it. And you can imagine the potential positives of this is you're trying to answer questions by this trial. We're asking Mother Nature questions. Does this drug work? Is this device safe? And during the course of the trial, if you're able to look at the data, you learn things that you didn't know when the trial started. You would have designed a different trial if you knew that. And so you start to learn that. So can you learn efficiently to make a better design to answer the questions more efficiently and maybe perhaps answer more questions within it? And you can imagine this can be a very, very powerful experiment to have this flexibility to it in that. So those are what adaptive designs are. Platform trials are a relatively new concept where you can imagine we run a lot of trials where, let's just take Alzheimer's, for example, where company A is running a trial of 500 patients on drug to 500 on placebo. Company B is running a trial of 500 on drug to 500 on placebo. Company C is running 500 on drug to 500 on placebo. As an ecosystem, we're enrolling 1,500 patients on placebo. And D's in A aren't used for B, and B's aren't used for A and all of that. So you bring them all into one trial. You enroll A of 500, B 500, C 500, and 500 placebos that they all share and compare to. We've just eliminated 1,000 placebos, 1,000 patients taking nothing to compare to these arms. By working together in a scenario like that, we can be already much more efficient and still each one of A, B, and C get exactly the power, the inferential strength they needed. But by working together as an ecosystem, we're doing much better. So this is a trial that had some initial designs. This was being done in cancer where multiple arms would come in, but it was really a pretty new type of design. And then COVID hit. And the pandemic hit. And so now we have many possible ways to treat COVID. This is separate from the vaccines. But to treat COVID, there's all kinds of repurposed drugs to this. Do we want to do antivirals, steroids, IL-6 receptor agonists, all kinds of things that we're interested in treatments? We don't have the ability in two ways to go out and start a new trial. It takes a year to start a trial, and we're enrolling, and we've got way more questions than we do ability to run trials. So lots of platform trials were created. The U.S. worked on Operation Warp Speed, where their active program were all platform trials where they had common controls and they brought multiple drugs in. This was done in recovery in the UK. This was done the Together trial. The Remap Cap trial was a global trial. It was this immediate, oh my God, this is the only way to answer this question in COVID. And they were adaptive. So they were doing analyses. Remap Cap was doing analyses largely every 2 weeks, saying, do we know the answer? Do we know the answer? Stop enrolling that drug. It doesn't work. Let's focus on this drug or that drug. So it was really an incredible opportunity where almost everything we learned about how to treat COVID came from platform trials. This incredibly efficient way to investigate multiple arms simultaneously, and they were adaptive. We stopped them when the drug wasn't working. We stopped them when the drug was working, and we announced to the world, if you could announce that steroids were benefiting patients a month earlier, you save thousands of lives. And so all of these things came together as a demonstration of how incredibly valuable all of these innovations in the experiments that we're using had in the treatment of COVID.
Damn. Okay. Yeah. This is super clear. Thanks, Scott.
Let me show you how to be a good Bayesian.